My name's Kylie Wong, and this is my cell diagram and organelles. First, we're going to be starting with our plasma membrane, also known as a cell membrane. Its functions is it creates structure to the cell while also protecting it. The membrane is semi-permeable, meaning it allows things to enter and leave. It also controls interactions and communications between other cells, and it is formed by two layers of phospholipids called the phospholipid bilayer. Moving to cytosol, which is going to be the blue color within the cell. Cytosol, or also known as cytoplasm, is the fluid that contains all the contents within the cell, and it is primarily made of water. Moving to the pink dot, that's going to be our nucleus. It is found in most cells. The nucleus is the control center of the cell, and it also contains DNA. Surrounding the nucleus would be our nuclear envelope. It is the bilayer membrane, and inside the bilayer membrane is the nucleoplasm and the nucleolus. The nucleolus is the organelle that produces ribosomes. Moving to our endoplasmic reticulum, there are two kinds of endoplasmic reticulum, rough and smooth. We're going to be starting with our rough one. It is continuous with the nuclear envelope, and it is named so because it contains ribosomes. So it's going to be all these little purple squiggles with the tiny dots on there. Those are going to be our ribosomes. Ribosomes are responsible for peptide and protein synthesis. Moving to the smooth endoplasmic reticulum, it is continuous with rough ER but does not contain ribosomes. Smooth ER detoxifies the cytoplasm. Our smooth is going to be those blue little oval-shaped blobs. And our ribosomes are going to be the purple little dots that are floating within the cell. Ribosomes read genetic material and produce peptides and proteins based on the specific segment of DNA code that it reads. They are small organelles produced by the nucleolus of the nucleus, and they are found in large numbers on the rough ER. So again, the small little purple dots. Moving to this pink flat stack, that's going to be our Golgi complex. It is a flat stack of cisternin. And its function is putting the finishing touches on peptide and protein synthesis, and also synthesizing carbohydrates. From that, those broken off pink circles are going to be our Golgi vesicles, which are finished proteins that are packed in Golgi vesicles and transported within the cytoplasm. Vesicles sometimes export products out of the cell membrane called exocytosis. Moving to lysosomes, those are going to be our green circles throughout the cell. Lysosomes are a type of Golgi vesicles that break down organelles and particles. They contain digestive enzymes to break down material they encounter. Next to those lysosomes, we have peroxisomes, which are going to be the red circles. Peroxisomes, they resemble lysosomes but are produced by endoplasmic reticulum. They detoxify the cell by producing hydrogen peroxide to break down particles. We have our mitochondria, which are going to be the orange peanut-shaped organelles. They are oval-shaped organelles, often called the powerhouse of the cell. They have many internal folds known as cristin. The folds increase the surface area of ATP production, and the mitochondria synthesize ATP using a variety of starting materials. From that, we have our last organelle, which is centrioles. They are paired organelles that are arranged perpendicular to each other. Each centriole contains numerous microtubules, and our microtubules are proteins that are responsible for moving and arranging the components within the cytosol during cell division.